Hello everyone, this is Mukesh Poddar from Raigarh Narayana branch. Today I am going to explain a very important topic of class 12 biology from human health and disease, that is AIDS. So let's start with a clinical case. Okay. So what happened? A 28-year-old man admitted in a hospital with some problem, with some symptoms. The symptoms were he was having recurrent fever, weight loss and frequent infection. Now he was treated multiple times, repeatedly he was treated, but still the problem was there. So doctor went for the diagnosis, and after lab reports, the lab report shows a significant reduction in the number of CD4 T helper cells. Now you think, what kind of disease it could be? What is the problem? Like, from which problem he was suffering from? So let's try to understand what was the problem. So here we are talking about the disease which is known as the AIDS. Let us try to understand the full form of AIDS. So the full form of AIDS is acquired. The full form of AIDS is acquired immunodeficiency syndrome. With the full form only, you will understand that what kind of problem it is, what kind of disease it is. It is saying that it is an acquired disease. Acquired means this disease is not congenital in nature. This disease occurs in the lifetime. It is a lifetime disease. Like, this disease is not congenital. NCERT says it is not congenital. It is not from by birth. It is immunodeficiency. Immunodeficiency means it will reduce the power of your immune system. Reduce the power of your immune system. Power means the defense mechanism. The intensity of the defense mechanism will get reduced. And the last term is syndrome. So if you think, what is the meaning of syndrome? So syndrome is, NCERT says, it is a collection of disease or collection of symptoms, we should say. Collection of symptoms. So it is a collection of symptoms. Next, what kind of pathogen? Which pathogen is causing this disease? Which pathogen is mainly causing this problem? So this problem is caused by a virus, and the name of the virus is HIV. Most of the time, what students get confused between the term HIV and AIDS. Like, I have seen that students, they don't know the difference between HIV and AIDS. So students just try to understand HIV means it is a name of a virus. Human immunodeficiency virus. Whereas AIDS is the name of the disease. AIDS is the name of the syndrome. It is the condition, okay, like related to which we are like studying today. And HIV is the name of the virus. The full form of this HIV is human immunodeficiency virus. So the name of the pathogen is HIV. You have to understand the meaning of pathogen is disease causing organism. Okay. Now, the first case which was seen for AIDS, it was in 1981 in USA. And if you talk about India, so the first case was in 1986 in Chennai. Okay. Now, coming to the next section. What kind of virus it is? Students, for your information, I must say, and it is very important question for your NEET as well as board point of view, that this HIV is a kind of retrovirus. It is a kind of retrovirus. Let us try to understand what kind of virus it is. It is a retrovirus. Now, in exams, definitely will get question why it is known as retrovirus, because it consists of an enzyme. The name of the enzyme is reverse transcriptase. This is the name of an enzyme. What does enzyme do? Why this enzyme is so special, and why just because of the presence of this enzyme, this particular virus is known as a retrovirus? What is the reason behind that? Let us try to understand. So first you have to know one thing, that HIV is having RNA genome, as you can see in the picture. HIV is having RNA as its genome. RNA genome. How many strands? Two strands you can see. One here, one here. So two RNA strands are there in HIV. So basically, HIV is a RNA virus. Are you getting my point? HIV is a RNA virus. So you have studied in one of the chapter that if you compare RNA and DNA, the RNA is very unstable and RNA get mutate very faster. So you can understand that how much dangerous this virus is, because it is very difficult for getting vaccine for this virus. Same happened in case of COVID-19 also. That was also a RNA virus. So it is also an RNA virus. Okay. Now this HIV is a RNA virus, and the reverse transcriptase which is present in this virus, this enzyme, what it does, it actually converts the RNA into DNA by the process known as reverse transcription. Reverse transcription. I think you know that what is transcription. We know that transcription is, DNA to RNA. DNA to RNA. Is it? But the reverse transcription is RNA to DNA. So this work is done by the enzyme reverse transcriptase. Now if you look at the figure, it will help you to understand one more thing, that a GP glycoprotein is written over there. Can you see? Glycoprotein. First point is about the virus structure. What is important for you to know? That it is an enveloped virus. And the second point, that this envelope consisting of a GP, glycoprotein, GP. And the name of this GP which is outside is known as GP120. Whereas if you see the inside portion which is the spike actually, and that particular portion is known as GP41. Okay, so these are the name of the glycoprotein. One is GP120 and another is GP41. At the same time, you can see here one capsid. So the capsid is basically proteinaceous in nature. You know, it is made up of protein. Reverse transcriptase is the name of the enzyme which is present in it, and the RNA virus. So it is RNA virus, so RNA is a genetic material here. Done. I hope till this portion you have understood, and I'm telling you, from this page only you will get very important question. Coming to the next one. Now, mode of transmission. The NCERT says that the transmission of HIV is because of the following reason. One is sexual reproduction with sexual relationship without protection. That means if someone is having sexual contact with HIV patient, this is sharing of needles. Second, the first is this one, second is sharing needles. What is the meaning of sharing needles? Why? When we will share the needle? Students, you have to understand, during the drug cases, intravenous injection, the drug abuser use same drugs they can use, and at the same time, sometime in the drug ka nasha, they never know which kind of needles they are using, which type of needles, or what infected needles they are using, or normal needles they are using. They don't know, because whenever people abuse drugs, they take drugs, they always, most of the time, they take in a group. So uska dost baitha hai, to uska dost agar wo drug le raha hai, from the same injection he will also take that. और अगर उसके दोस्त को infection होता है, in that case he will also get contaminated, or he will also get the infection in his blood, in his or her blood. So this is one of the method. Third is the blood contact, means blood transfusion. If you take some blood from a person who is unhealthy, who is having the HIV infection in his blood, it can happen. Fourth, vertical transmission, and it is very, very important. NCERT has not used this term, but this is important. Through breastfeeding, through placenta, or through childbirth. Okay, this is the method by which it can be transferred. So these are the four mode of transmission. Now you have to understand how many fluids are involved. First fluid which is involved is blood. So it can transfer through blood. Next fluid you can see, it is semen, that is sperm. Third, you can understand one thing, that CSF could be that fluid. CSF, cerebrospinal fluid. But students, don't get confused. Sometimes some students says saliva. I'm telling you, saliva cannot transfer HIV. Okay, saliva cannot transfer HIV. Till today, no cases of HIV has been reported through saliva. Now, Up to here we have understood how it transmits. Now we have to understand that how it works in the body. So inside the body, how it works. You see, the retrovirus is written over there. You see, the retrovirus is written over there. Let's try to erase it. Retrovirus is written over there. Sure. This retrovirus is having viral coat protein. Now this RNA virus is trying to enter into a cell. This name of the cell is macrophage. Macrophage. Macrophage is the name of the cell. So when it tries to enter inside this viral RNA is introduced into the cell. After that, what will happen? This RNA will be converted into DNA with the help of the same enzyme, reverse transcriptase, and the name of the process is reverse transcription. Clear? Now, after the conversion into DNA, this DNA will incorporate into the host genome with the help of an enzyme which is known as integrase. It is not mentioned in your NCERT, but it is important. With the help of an integrase enzyme, this will enter inside the host genome. After that, what will happen? The host genome will get transcribed, it will get translated, and after translation, what we will get? Obviously, protein we will get. So this HIV protein, this HIV protein will be produced, and the entire HIV virus will be manufactured inside the macrophage. And that is the reason NCERT says that HIV factory is macrophage. This is very important question. Name the cell which or cell organelle or cell which can be considered to be HIV factory. So macrophage is considered here as HIV factory. Okay, so many HIV cells are produced. Now what will happen when the macrophage cells get destroyed after that, or like it will act as a HIV factory? What will happen after that? So after that, what you will see is the macrophage is acting as HIV factory. That we have understood. It is acting as HIV factory. After the HIV factory or after the macrophage attack, these virus will start attacking simultaneously, not after, simultaneously, they will start attack the T helper cell. Student, here I want you to focus because this portion is very important. They are attacking the T helper cell. So ultimately the T helper cell amount will reduce. So are you getting my point? Why that blood report was showing less number or significant reduction in the T helper cell? So the T helper cell is getting reduced. Next, what will happen? When the T helper cell will get reduced, during that time, the cell-mediated immunity will get hampered. Is it so? Now this, just because of this cell-mediated immunity, what will happen? The opportunistic infections or opportunistic microorganisms like toxoplasma, mycobacterium, these diseases, like these disease-causing organisms, which otherwise can, like body can otherwise can be, like can prevent it, like will not allow it to infect our body. But during this case, when the cell-mediated immunity get compromised, during that case, what will happen? There will be case of opportunistic infection. And just because of the opportunistic infection, the person will get sick again and again. So, which bacteria can cause problem that time? Mycobacterium can do problem. According to the NCERT data, toxoplasma, they can do the problem. So these are the opportunistic infection which are actually occurs that time multiple times. And this is very, I am telling you, this is very, very important. Which type of immunity get compromised? Cell-mediated immunity. And the amount of T helper cell will reduce significantly. Okay, number of T helper cell will reduce significantly, and that is the reason why our body immune system get deficient. Clear? So macrophage will act as a HIV factory, and T helper cell will, the amount of T helper cell will get reduced. Now coming to the diagnosis. That patient who was admitted in the hospital, what do you think? How he was diagnosed? So he was diagnosed with a very, very important method that is known as ELISA. ELISA. The full form of ELISA is enzyme linked sorbent assay. And I'm telling you, this method is very important because it is based on, like examiner can ask you, it is based on antigen antibody reaction. It is based on antigen and antibody reaction. Okay, so this is very important method for identification. Confirmatory test is there. One confirmatory test is there. Confirmatory test, that is Western blotting. Western blotting is considered to be the confirmatory test. Okay, confirmatory test is Western blotting. So this is all about the diagnosis. Now coming to the treatment. Now if somebody asks you what is the treatment for this HIV or AIDS, so I'm telling you there is no permanent treatment which can cure this problem. Okay, so how it can be done? So what data we are having? We are having few drugs which are known as ART. The full form of ART is antiretroviral drug. What this drug does, you know, NCERT says that death is inevitable, like marna to hai hi, but we can prolong the life. How we can prolong the life? We can give this ART. What this ART will do, example azithromycin, the name you can see, this is the name of a brand or the name of the medicine. What this medicine will do, you know, this medicine will reduce the load of virus, this medicine will reduce the load of virus. How? By preventing the multiplication step, multiplication or RNA elongation, or you can say multiplication of the RNA of this virus, and that is how they can stop the growth. They can help for temporary like stoppage of the growth, but multiplication will definitely occur, and at the end of the day the life can be prolonged but person will definitely die. So this is ART which can be used. Now let's come to the prevention, and it is very important. Don't die of ignorance. During like 1938 or 1981, sorry, 1981 and 1986, during that time this statement was given by UK government that don't die of ignorance. Students, you have to understand that this disease occurs because of the human behavior which is very ignoring type, which is very casual type, like they have multiple sex partners, they don't use contraceptive methods, they are not concerned about their health, and if they are having some symptoms also, they don't go to doctors very quickly because going to doctor is a big headache for everyone. Now this statement is very important, that don't die of ignorance. We have to educate people. Like various organizations are there, NGOs, non-governmental organizations are there, which are helping people to understand that prevention is better than cure. And I'm telling you, there is no permanent cure for HIV AIDS. So it's better, we should prevent it, and we should not ignore like fevers also. If the fever is recurring continuously, if the fever is becoming a chronic case, you should not ignore it. Okay, so that is very important. Few agencies are there like NACO, National AIDS Control Organization, and some NGOs are there who are working. We are also maintaining World's AIDS Day to educate people. We are using different print media, we are using different days, and hospitals are also distributing condoms free of cost. Why? So that people can use it multiple times and they can get safe from this sexually transmitted disease. You have to understand, in conclusion, what kind of disease it is. It is a sexually transmitted disease. Okay, it is a kind of sexually transmitted disease. So don't die of ignorance. That's all for the concept part. Now let's turn back to the question answers part. Practice session. Let's try to understand and let's try to solve this. Okay. First question. AIDS is caused by HIV. Among the following, which one is not the mode of transmission of HIV? Transfusion of contaminated blood, sharing the infected needles, shaking hands with the infected person, sexual contact with the infected person. What will be the answer? Just pause it and tell me the answer. Okay, so the answer is shaking hands with infected person. Just by shaking hands with the infected person or sharing the food or like hugging with someone, kissing someone will not have any issue. AIDS will not communicate like that. The early screening test for HIV infection is ELISA, Western blot, culture of HIV, detection of opportunistic infection. So we have studied that ELISA is the primary way and Western blotting is the confirmatory test. Coming to the next one, opportunistic infection appears in AIDS patient when the HIV has destroyed. Opportunistic, I said opportunistic infection. When it will occur? When it will destroy macrophage, T cytotoxic, T helper or B cell. So we have seen it is T helper cell. During that time, Toxoplasma gonadie as well as the different kind of bacteria like Mycobacterium can affect and it can cause problem. At which stage of HIV infection does one usually show symptom of AIDS? AIDS symptom. When viral DNA is produced by reverse transcriptase, when HIV replicates rapidly in the T lymphocyte within 15 days of sexual contact, when the infection this point is very important, I must say. This I missed, I think. When the T lymphocyte, T helper cell get reduced, during that time the person will suffer from bouts of fever. He or she can have vomiting also. Okay, this kind of problem can be seen. Coming to the next part, study the diagram given and choose the incorrect statement. So out of that you have to identify incorrect statement. Here the A is showing retrovirus replicates outside the cell. No, it is incorrect. It is incorrect. So viral DNA is produced by the host enzyme. Yes, viral DNA is produced. Next, viral DNA is incorporated into the host enzyme. It is also correct. So the correct answer is this one. Next, HIV that causes AIDS first starts destroying which cell? Obviously helper T lymphocyte. Same question. Next, complete the analogy by choosing the correct option. Typhoid is of Vidal test, so HIV is of which test? ELISA. Next, the letter T in T lymphocyte refers to as, students, this T refers to the thymus. T refers to thymus where the T lymphocyte actually matures. Okay, so that's all for today. Thank you so much. Mukesh for the signing off.